Hello, biology, and welcome to the last part of Chapter 8, 8.3, where the topic today is about meiosis. Meiosis is going to be the last of the cellular reproduction methods that we're going to talk about, and this is one that we're only going to see once again in eukaryotes. So, meiosis, the whole point of meiosis is to divide the cell in a way that reduces the number of chromosomes by half. So in humans, we start out with 46 chromosomes. By the end of meiosis, we're going to have cells that have 23 chromosomes. And this is used to create the gametes, or the sex cells, egg and sperm, is the point of meiosis. Alright, so cells prepare for meiosis the same way that they prepare for mitosis. They undergo the normal G1, the gap1 phase, the synthesis phase, and the G2 phase. Because they need to do this because they need double the organelles, they need double the DNA, they need double the cytoplasm. So, just like in mitosis, they start out with double the chromosomes. Now, the difference with meiosis and mitosis, the cells split once to create two cells. In meiosis, these cells are going to split twice to produce 4 haploid cells. So one cell is going to split twice. And it happens in 2 stages, one we call meiosis 1 and one we call meiosis 2. Just like in the mitosis one, there's a walkthrough for this one. If it has the star, it should go on your walkthrough. Make sure you draw the pictures. They will help you immensely moving forward. Alright, so prophase 1. And all of the meiosis 1 is just going to have a prophase, metaphase, anaphase, telophase, cytokinesis. They all have that one designation. And then the second part will be meiosis 2. So I have on here that this in interphase here, these chromosomes are hanging out. They have doubled up here. You can see that there's this big X's here now. So they're doubled through the S phase in between interphase and prophase here. But in prophase 1, this DNA tightly coils into the chromosomes we see. The spindle fibers appear. The nuclear membrane disappears. And we get this synapsis. And what synapsis is instead of getting we get these homologous chromosomes to pair up. And they form these tetrad because there's 1, 2, 3, 4 copies of those chromosomes. So there's 2 copies of mom's chromosome and 2 copies of dad's chromosome. Chromosomes 1 from mom and dad will pair up, form a tetrad in prophase 1. Because of this, we get some brand new stuff, some brand new genes and mixture of genes that have never happened before. So randomly during this synapsis, chromatids from the homologous pair will twist around each other, parts will break off, and they will reattach. So as you can see, these new chromatids here are completely new. It's mostly dad's DNA here, but it's got a little bit of this mom DNA here. This is called genetic recombination, and this is a mixture of genetic material that has never occurred before in the human population. That's the really big perk to this meiotic process. Brand new process here. This is why siblings don't look the same. This is why we have so many different human traits, is because of this process here. And this is during prophase 1. Alright, metaphase 1. In metaphase 1, just like in metaphase of mitosis, these tetrads are going to line up at the middle here. But they're going to line up in pairs instead of all the way stretched out. So before, these 2 chromosomes would have been on top of each other and not attached. Right now, they're going to be in pairs. So there's going to be 23 pairs of chromosomes on the line here. They'll have spindle fibers attached to the centromere, and then those will start to pull apart in anaphase. So anaphase 1. These guys are going to move towards the different poles of that forming cell. So one of the homologous chromosomes moves to the one pole, one moves to the opposite pole. This happens randomly. It might take one chromosome from mom here, but the next one up might be from dad, from mom, from dad, from mom, dad, 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 mom, mom, mom. This is called independent assortment, and this random separation of these homologous chromosomes. There's no rhyme or reason to why they do it. All right, telophase 1 and cytokinesis 1 are on here. This is the telophase 1, this is the cytokinesis. In telophase, the chromosomes reach opposite ends, and the cytokinesis begins. And for cytokinesis, it's important to note that these cells are haploid. They have 2 copies of each chromosome, but they don't have both homologous chromosomes. So this is just the mom chromosome on this side, and this is just the dad chromosome on this side. There's 2 copies of them, but for chromosome number 2, let's say this is chromosome 2 that we're talking about, now each cell only has 1 copy of it. That means that it's haploid. And this is all because of that cell going through the S phase before meiosis to double up these chromosomes so that we can have this structure here. All right, meiosis 2. So we don't copy the DNA before the stage. The cell doesn't go out of the process. It starts this immediately after cytokinesis 1. But it's important to right now, each cell has 2 copies of half of the genome. All right, prophase 2. The spindle fibers form again, move the chromosomes toward the midline, and metaphase 2, they're in the middle here. They're lined up. And Anaphase 2, they're starting to separate, getting pulled from the tails here. So now these chromatids are getting separated. In Telophase 2, the nuclear membrane forms around these guys, around the chromosomes of the 4 new cells, and then cytokinesis, these cells chop in half, and we get 4 cells at the end of this. And these 4 cells are all 1N, or they have half of the chromosomes. So all of these have 23 chromosomes that have been randomly chosen from mom and dad. In boys, we go through the process of spermatogenesis, where this formation of sperm cells happens. It starts from 1 cell that goes into 4 sperm, and right along the line, before they become mature, they become spermatids, which are these immature sperm cells. They will mature later and then be ready for reproduction. But in males, all 4 of these cells are viable to create young. It's going to be a little bit different from females, but in males, meiosis makes 4 sex cells. All right, oogenesis. This is the production of mature egg cells, or ova. And oogenesis is going to take 1 cell and create only 1 mature egg cell, 1 ovum. So when cytokinesis happens in all of these, there is a unequal division of the cytoplasm between the new cells. The ovum gets the majority of it. The other 3 become what we call polar bodies. And these polar bodies eventually are disintegrated. They're not viable for reproduction. Only this 1 ovum is viable for reproduction. So in male, you make 4 viable sex cells. In females, you make 1 per round of meiosis. All right, as organisms, there is some perks to sexual reproduction. The perks of this are there's a lot of different genetic variability that happens because of this. Siblings are very different from one another. Cousins are very different from one another. As you go farther and farther back, once you get to 4th, 5th cousins, they start to be completely different. This enables our species to adapt very quickly, to get new genes, to get lots of mutations like red hair and blue eyes, and to continue to get these new adaptations as a species. For other species that also sexually reproduce, this is the same thing. They are able to adapt quickly to their environment. They're able to get new genetics. Next chapter, we're going to talk about how that actually works. We're going to start talking about mutations, and we're going to start talking about alleles and traits. But this is the end of chapter 8. I hope that you now understand the process of meiosis. If you have any questions, let me know, and I'll see you in the next one.